Okay, so we're trying to navigate between speaking to many people online and some people in the room, so we're gonna make it work. So I'm Tamar. Good afternoon to everyone. I'm gonna start with a little bit about a little story about myself and how did I get to this line of research. And I am trained as a child psychiatrist. After my residency, I spent 2 years as an inpatient in an inpatient unit as an attending physician, and there I would treat kids with severe psychiatric conditions, usually in their first psychotic episode, which is a very extreme mental health condition. And if you know that, you're back. It's an extreme mental health condition, and there's a lot of work. The days go by really, really fast. You give a lot of medications. You support the child and the family, but day in and day out, I started to feel that I'm treating conditions that I don't really know what's going on. And everywhere I look, heterogeneity was around. I was looking at I was looking at the literature. We used to say that like schizophrenia is many conditions, and each child is very different, and it's very true. Until one day, a child with 22q deletion syndrome arrived to the department or to the unit, and she had a deletion in chromosome 22, and I learned that that puts her at an increased risk for schizophrenia, for psychotic conditions. And that basically opened up a window to a line of research that I was not familiar with, that's called genetic first or neurobehavioral psychiatry. And this is basically what we're going to talk about today, about genetic first approach in psychiatry. I'm going to talk a little bit about 22q deletion syndrome, but I'm mostly going to talk about our current work, which is focusing on oncogenic pathways, pathway, the MAPK pathway. And I will try to convince you that it would provide very valuable cues or clues to the epigenetic, to the etiology of neuropsychiatric. So we're going to start with a framework. So the framework of this condition and some kind of general information about the field is that when we talk about rare genetic conditions, we can talk in a very general terms on two type of mutations. One going to be a single gene disorder, and how you can see on the y-axis that single gene disorder has usually a high penetrance, meaning that if you have the mutation, you have the phenotype, and but they are not very common in the population. Versus aneuploidies or copy number variation of chunks of the genes or chunks of the chromosome or the chromosome themselves, which are more frequent in the population, but their penetrance is lower. And what you can also see on your left side when we talk about aneuploidies or we talk about copy number variation, and you see this polar graph, you can see that we can basically estimate the odds ratio of the effect of different duplications or deletion, because in this gene variation, we can have a duplication of the chunk of the chromosome or a deletion of a chunk of the chromosome. Each one of them convey a different risk. Many of them convey risk for ASD, for autism. Some of them will convey risk for schizophrenia. However, when we go to this region of single gene disorder, there we have a gap in our knowledge. We can't really estimate the odds ratio because we don't have enough controls. These conditions are, at least the ones that were reviewed here, are very rare, so it's difficult to get to this data. And a little bit more about this framework and how we think about it. So as I mentioned, for the same region in the chromosome, there can be a deletion and duplication. I will use 22q deletion and duplication to show you that we can contrast between them. And if we contrast between them, there are some symptoms that we'll see present in both of deletion or the duplication. For example, in this case, anxiety in green. But there are some cognitive phenotypes or psychiatric phenotype. In this case, for example, verbal IQ in red, that is expressed differently based on if we have haploinsufficiency of these genes, meaning that we have a deletion or we have a duplication. From more recent work from my lab, you can, what we did here, this work focused on aneuploidies, meaning deletion or absence or presence of X chromosome. In this case, it's X chromosome aneuploidies, and we tested two conditions. One is Klinefelter, when we have XXY and the phenotype is male, or we have X0, which is Turner syndrome, and the phenotype is female. But contrasting them do allow us to ask the question whether an excess or absence of the X chromosome affect the phenotype in any way. And for example, here we can, I was very interested in attention and in executive function in this condition because we know these conditions are prone to these phenotypes. So you can see that, for example, both phenotypes, both Turner syndrome and Klinefelter have, these are effect sizes. They have an effect on their auditory attention span, but only Turner syndrome will have an issue with inhibition. So we learned that the absence of X chromosome affect more specifically inhibition. And to take that a further step, we can not only ask questions about the behavioral or cognitive phenotype, we can also ask questions about the brain. And here I want to also talk about a different concept. Not only we know there's effect of these duplications and deletions on the brain, we also, if we contrast the effect size of how much those syndromes affect the brain, we see that the effect sizes are typically much larger compared to effect sizes of idiopathic condition like bipolar disorder. Schizophrenia is actually one of the bigger ones. Mood disorders, and more specifically, the disorder that I'm concentrated in are ADHD and ASD. So this is just to show that these conditions are with large effect size, which allow us to ask questions and have kind of like an access to ask questions about mechanisms that we typically, when it's very heterogenic, we can. And more specifically to our talk today, I'll talk about a syndrome called Noonan syndrome. I'll explain in a second what it is, but the main goal of this slide is to show you that also for Noonan syndrome is the effect size on, in this case, subcorticals, it's large. The subcorticals and specifically the striatum are shrinked in Noonan syndrome, and this is the effect is much larger when we compare the effect of ADHD or ASD on those same structures. Sure. Yeah. The cortical volume is also decreased, but when you show this, when you look at this data, both for the work in ADHD, ASD, and Noonan, we always control for the total brain volume, because otherwise it's like, you know, it's not fair. So yeah, it's after controlling for them. And more specifically about the pathway and about the oncogenic pathway, and why do I call it an oncogenic pathway? The MAPK pathway, this is the pathway we'll talk about today. And this pathway is very strongly related to human cancer. And the way it's related to human cancer, it's by somatic mutation. If we have mutations along this pathway, along genes that encode proteins on this pathway, this mutation has been associated with colorectal cancer, pancreatic cancer, and many more cancers. It means that the scientific community phenotyped this pathway really, really well, because we already have medications that can intervene with this pathway, and this medication will be medication that used in oncology. But these mutations are somatic, meaning that can happen during life to each one, each individual. But there are some kids that are born with a germline mutation, meaning the mutation happened in the sperm or the egg, and they are born with a mutation that basically happens in orbital cells, right? And the two specific syndromes that I want to emphasize today, one of them is Noonan syndrome. These are the kids you see here, and one of them is neurofibromatosis. I'm going to talk more about Noonan syndrome today. And to convince you that this pathway is not only related to oncology, but also related to psychiatry and specific neuropsychiatric conditions, I kind of going to demonstrate it in several ways. One way would be what I did here is I surveyed this database, which is the ASD Safari gene database, and in this database we have lists of genes with high confidence or suggestive evidence for association or being enriched in kids with ASD. And as you can see here, many genes on the pathway are being implicated in this condition. So everything you see in orange or in yellow is part of this pathway. For example, I talked about SYNGAP, for example. It's a very has a strong evidence to be related with syndromic ASD, but also NF1 that you can see here, and PTPN11 that you can see here. And the reason that some of them are green and some of them are green and some of them are red is because those genes can be either inhibitory or encode genes that activates the pathway. So the inhibitory genes, if they're affected, for example, NF1, it's an inhibitory gene. When there's a mutation, the mutation inactivates or reduce the amount of the protein, and then because it has an inhibitory effect, the pathway will work in axis. But similarly, when the genes that are mutated, the gene in green, when they are mutated, they actually gain function, so they also activate the pathway. So basically, in whether it's a red or green gene, colored in red or green, it's always an activation of this pathway. Similarly, as far as I know, in oncogenic condition. And it makes sense because the pathway is responsible on, it basically connects its signal between the cell to the nucleus, and it relates to transcription, so, and proliferations of cells. So it makes sense that it's going to be related to pathology. So to continue and think about the association between psychiatric conditions, specifically ASD, and this pathway, there's other evidence. This is a recent work that used a combination of methods and identified 72 new genes. And among them, as you can see, 3 of them, again, the SYNGAP, NF1, and here at the end, are genes that are part of the MAPK pathway. In a different work from last year, the researchers were able to identify 6 archetype pathways that involve in ASD, working on large data set of 42,000 kids. And the RAS-MAPK pathway, again, emerged as a pathway that is implicated in AIDS. And lastly, this is a shout out to Laramie Duncan. In collaboration with her, we just surveyed GWASs, the recent GWASs, and just looked on the RAS-MAPK pathway genes. Although you can see that the association of these genes with ADHD in this case does not reach the level of significance that you need in GWASs, but it still provides some evidence of the involvement of these genes. So you can see that it's true for ADHD, for autism, I was surprised by the bipolar disorder and schizophrenia because more gene emerged there. And major depression. And here in schizophrenia, it's actually SYNGAP and RASA3 are strongly associated. So this is kind of like a general evidence for the involvement of this pathway in neuropsychiatry. And now we're going to dive into the actual work with the kids. Are we good? Yeah, okay. So we're going to dive into what we actually do in the lab. And we're going to focus on, as I said, on Noonan Syndrome. And Noonan Syndrome is caused by 75% of the cases caused by PTPN11, which is the most common gene that causes Noonan Syndrome, 50%. And between 10 to 15% is caused by SOS1. Both of them encode on the RAS-MAPK pathway, cause hyperactivation. There are other genes, but we're not gonna focus on them today. This is the most common, what we call rosopathy, a syndrome that is associated with the RAS-MAPK pathway. The IQ is in the normal range, and this is important to us when we think about the inferences we can make related to neurodevelopmental conditions, because once there is a strong association with lower IQ, only by the lower IQ we will see more neuropsychiatric manifestations. And our work with that cohort, which included 45 kids aged 4 to 11, first step, we just phenotype them in terms of psychiatric manifestation. We administer the KTAT. And as you can see, we identify high percentage of ADHD, autism spectrum symptoms. None of them fulfill the full criteria for autism, but many of them presented with symptoms and an oppositional defiant disorder that I think is very related to irritability, but that's kind of like a future work. And we not only focused on the dichotomy of the DSM, but we also wanted to look on the dimensions. And we looked on the dimensions we kind of like suspected that related to ASD, ADHD. So we look on attention and executive function, we looked on social skills, and we looked on aggression. And I was afraid it wouldn't work. And you can see the results here. I'm happy to kind of like spend more time, if you like, in the Q&A, but I want to say that generally we saw in these 3 domains that kids with Noonan syndrome presented with higher symptoms profile almost across the board of attention and executive functions, social skills, and aggression. What I want to emphasize here is that the significance for most of them, except from the one that you see NS, stayed also after we controlled for IQ. So So it kind of tells us that the IQ is not a confounder here in the elevated symptoms that you see. For some cognitive measures, we lost the significance. My thought is that we lost the significance because the variance being, we're basically testing the same things, so it's kind of expected. So we talked about the behavioral phenotype, the cognitive phenotype. We talked about the genetics, the proteins, and the cellular. But early on, we identified that there's a gap in the knowledge, and we basically didn't know anything about the brain of the kids with Noonan syndrome, and this is where the work went. And I know it's a mixed audience, so I'm just going to give a 101 really quick intro for brain imaging, so you can understand what I'm saying. And again, happy to dive more into that. So I'm going to present work actually more from structural imaging, and basically what we do, we take like an image of the brain, and we then use different methods, but in this case, we parcellate the brain to regions, and then we have an ability to measure the volume of those regions, the thickness of this region, and the surface area of this region. And we also have ways to look on white matter, which is the connection between these regions, if we want to be very simplified. And in the first, at the beginning of the work, we had only 12 kids, and we wanted to choose who we're going to focus on, because we have a problem of multiple comparison in imaging, right? If we test everything, we're just going to find a significance. So we want to focus on one hypothesis, and our hypothesis, because of the high association with attention symptoms, we want to focus on the striatum. And the striatum is, and I just want to kind of like emphasize that the striatum that we are measuring with our tools currently is mostly the dorsal striatum, and the dorsal striatum is responsible for function selection, planning, cognitive flexibility, response inhibition, working on a lot of the executive functions. So just to put that in context. And we did an initial work with 12 kids, as I said, and we published that in 2017 that shows that these regions are shrinked. And then we repeated that work with 40 kids. And this is the results you see here. And basically what we found is that the QDET, the putamen, and the pallidum are significantly smaller in kids with Noonan syndrome compared to controls. But we also were able to divide it to kids with PTPN11 and SOS1, and then we start to see that there's also some differences and some gradual effect between the mutations. And what you see here is an illustration of the effect sizes from light blue, effect size of 0.2 up to 0.1. It's a minus, just to emphasize that it's smaller and not bigger. And you see the effect in PTPN11 that is stronger than SOS1, but both of them basically affect the striatum in the same direction. This is just another visualization of what I just showed you, and we have a way to test the significance of our effect sizes. So you see that PTPN11 consistently affect the striatum, and SOS1 is kind of like have an effect, but not as significant. And we also see some specificity in terms of like it affects the striatum and does not affect other subcortical structure. And to finalize that, we wanted to test whether it's associated with behavior, whether the volume are associated with behavior. And on the x-axis, you see the volumes, and on the y-axis, you see the function or the function on these two tests, the attention, auditory attention and inhibition, that we know that are affected in Noonan syndrome. In red, you see the strong correlation of Noonan syndrome with in the Noonan syndrome group between the brain's volume, the striatum volume, and the cognitive function. But as you might notice, it's actually in the different direction than we expect, because smaller structures, kids with smaller structure actually function better. So this is something that we cannot explain. And finally, Karen, you asked me about effect on cortical regions. So at N of 40, we were able to test that, and we did find effect also on cortical regions, mostly on temporal regions and parahippocampal regions, what you can see here. And what we then did, we used data sets from the Allen Brain Institute that basically give you expression of different genes, and we took the PTPN11 expression levels, and we just correlated that with the effect sizes of our surface area, because we saw it affected here. And again, we found actually a positive correlation between, in each region, how much the PTPN11 is expressed in the typically developing brain in Allen Institute, which are adults. And the effect we find on in our cohort of children with Noonan syndrome that have an axis activation of this pathway and this gene. And this basically provide us some evidence that is outside of the Noonan syndrome itself. So it's a step towards the generalization of the finding. I want to see how. Okay, perfect. So this is one work we talked about, and now I'm going to talk about a second one, and then we kind of are going to conclude. So we were already able to show that the phenotype and the cognitive aspect of Noonan syndrome is affected by the syndrome. We were also able to show that brain structure is affected. Data I didn't show you, but is already published, is that we also showed that there is an effect on white matter. But then we wanted to look at function, brain function. And that was the next step. And to explain that it's not the same thing, right? You can have a structure of your brain, but then it functions differently. Of course, there's a strong relationship between structure and function, but it's not the same thing. So it is very important to ask questions also about the function of the brain. And a little bit of background to understand the work. So we are looking on resting states. Resting state, it means that the child lies in the scanner and does not have a task. And this work is based on this seminal work that the way it went is that that was a task-based. That was a task-based functional MRI, and basically the participant was asked to push to do some model task, and then their motor cortex light up in the scanner. But what they found out is that when they analyzed the brains while the person was in the scanner but not doing the task, basically resting, they found that if they put a seed here and look at the rest of the brain, what actually lights up, it's the same circuit. So from there, maybe you heard the fire together. So neurons and circuits that usually work together, they will fire together, and we can, with resting state, identify those signals. So we did two things. One analysis was a seed-based analysis, similar to what I just explained, except instead of putting the seed on the motor cortex, we put the seed in the striatum, because we had evidence from structure that it's affected, and we had evidence that is also related to phenotypes. So when we put the seed here in the Q-date, we ask which regions of the brain are connected to that and are differently connected between our Noonan syndrome and our control. So we saw that there's hyperactivation here and ipsilateral, right, on the other side, and hypoactivation with frontal regions. Similarly, we did the same thing in the putamen, and we found these connectivity patterns basically more on the subcorticals. And then we did a more general analysis, looking at the whole brain and letting it's more data-driven approach that we apply ICA analysis and we identify components of the brain that work together. So first step, we identified that we can produce in our data networks that we know, and we know that they're robustly identified in other regions, because when you do it for the first time in a syndrome, you first want to show that you actually asking question about something that is relevant, and not that your syndrome is like completely function differently, which is not expected, but it's just kind of like a sanity check for us. So this was the first step. And the second step was to compare these networks and see if they more activated or less activated in Noonan syndrome. And these networks that you see here, basically the ventral attention network, the frontoparietal network, visual network, and subcortical network, or limbic network, were more activated. There was hyperactivity in those children with syndrome. And to finalize this work and trying to think, okay, what does it mean? And if it's related to the behavior of the kid, we also did again a brain behavior correlation, but this time we wanted to do it more in a data-driven way. We didn't want to decide, okay, it's going to be attention or it's going to be something else. We wanted to see what the data basically tells us. So we ran on a very rich, what we're doing with the kids, maybe I needed to say that at the beginning, we're doing a very deep phenotyping. So we're doing psychiatric disorder phenotype, but we also run a lot of cognitive testing and a lot of, we collect a lot of data on each child. So what we did with this behavioral and cognitive data, we put that through a principal component analysis. We came up with 11 components, but 3 of them explain 80% of the data. So we took the 3 first principal components. And then we looked at each component and decided how to kind of like call it, or how to, what is more contributing to that component. And then we ran the correlation with the brain. So what I'm showing you now is only the correlations that were significant. And the only principal component that was significantly correlated with the brain was the inhibition motor component. These are the components that contributed to this component. And when we ran correlation between this principal component, score for each person on this principal component, and with limbic and frontoparietal region, we found a positive correlation. So the more active the brain, the child is with Noonan syndrome is functioning better, which is also counterintuitive, because if hyperconnectivity is related to Noonan syndrome, and kind of like you think if hyperconnectivity is more related to more child that is more affected, you would expect the other way. So the direction of our correlation are not very clear to us. We have some hypotheses, but I do want to kind of like leave it open because it's only what we think about it. We don't actually know. And here I'm just going to summarize and conclude and leave you with some thoughts. Is that I wanted to show you that disease gene in RAS-MAPK pathway are involved in human neuropsychiatric conditions, and we found that they affect the striatum, but they also, when we have bigger cohort, we are now able to show that they affect also the surface area and the cortical thickness of the brain in a specific way, and that in brain function we find that these regions, this Noonan syndrome can affect 4 networks. 2 of them are related to attention and executive function, the frontoparietal and the attention network, and collectively it shows that we can definitely delineate connection between activation of one gene on the brain structure and function, and the actual phenotype. And what's very, very exciting about this type of work is that many times you write, like in a grant, this can serve as a target for like brain manifestation can serve as a biomarker for intervention. But in the case of the RAS-MAPK pathway, we already have interventions, and they're already being given to kids, because these kids, kids with neurofibromatosis that have tumors, benign tumors on their skin, already get these medications that basically inhibit the RAS-MAPK pathway. They're called MEK inhibitors, and it's kind of like already there. And the question is whether this inhibition of the pathway will help with modifying the neuropsychiatric manifestation, because many times you'll ask parents, and after the child is over the threats of cardiac issues or other issues that they're born with because of the syndrome, what's limiting their function eventually is the social difficulties and the executive function difficulties, the difficulties at school. So this is actually really important to parents and families. And just want to say thank you, and thank you for my team that is here also. And give a shout out that we are recruiting a postdoc, and thank you for my funding, MCHRI. That's it.
Fantastic. If folks have questions in the room or online, we've also encouraged our online audience to unmute and go ahead and ask any questions. Anyone in person?
Yes. Hold on.
Oh. Hi, very fascinating talk about the oncogenic pathways and those role in neuropsychiatric disorders. I'm curious if they share the same mutations or genetic variation. So you said the oncogenic pathways in cancer arise from somatic mutations, whereas these arise from germline mutations. So is there a higher incidence of cancer in patients with psychiatric disorders?
I will answer that.
And the other way around too, right? And the other way around, if you have a cancer as a child, or you
Okay, I'll answer both. So for the first question, whether these kids are more prone to tumors or malignancies, yes. Yes, it depends which syndrome. The neurofibromatosis one, basically NF1 is called NF1 cancer gene or something. It's definitely a gene when it's mutated, even germline. It's associated with mostly benign tumors, but these kids are at higher risk for gliomas. For example, for us, when we scan the kids, so each scan is being read by a radiologist because we do know that we scan a population that is an increased risk for cancer. Yes, so that's the answer. Yes, there is something very interesting about it that is more related to Noonan syndrome, because Noonan syndrome, they have like slightly increased risk for leukemia, but which is not like in NF1. It's less. And when this mutation happens somatically, they cause cancer for sure. But when you compare how much the mutation, specific mutation, the PTPN11 gene is big one, and when you compare the mutation that cause malignancies in somatic conditions compared to the mutation that happen in germline and syndromatic, they don't activate the MAPK pathway to the same amount. So the oncogenic, the cancer mutation that happens somatically activates the pathway much more. Because you can measure that, you can measure you basically can measure the product. And so you can measure the ERK phosphorylation, so you can show that those syndromes. And for the other way around, I'm actually not an expert. I think there's a lot of association and work in this field, but I'm not very familiar with it. Yeah. Yeah.
Hi, I'm Yafa. I'm a postdoc in Tamar's lab. Okay, so I have two questions. One about inhibition. Like you have been like telling a lot about like how inhibition is like not so good with the children with Noonan, and I have like I have a curiosity if it's something that happens in all of CNVs and all of genetic syndromes, or is something more specific to only. So I think that will be like interesting to I don't know to think about. And the second one is if you already know that like the patients with Noonan are already taking the meds, if you have thought or you have been working with the oncologist to start like just taking cognition and before and after and also
Thank you. So for the first question, I think inhibition is affected in many of the syndromes. The earlier work that I showed in aneuploidies was that we do see that some syndrome are more affected and syndrome are less affected. And I think it's inhibition and all these phenotype, behavioral cognitive phenotypes, they are the end product of many processes, so they're going to be affected. But it is, we have some work comparing girls with Noonan syndrome to girls with Turner syndrome. So some of those phenotypes are very similar, but some of them are not. So I think there's some specificity there. And the kids that are getting the MEK inhibitors currently are kids that have NF1 with plexiform neurofibromas. And there is one researcher that kind of catch them before and after. But the results are very weak. I think it's because of how it's built, because cognitive and cognitive ability is not going to improve just by changing the pathway activation. It requires, it's like a muscle. Like you can give something that will allow the muscle to develop, but then you have to develop it. Otherwise, it's not going to happen. So I think the structure of these studies needs to be different. And there are some medication, and MEK inhibitors are actually not that. But it is a it's a type of chemotherapy, and it's not really kind of like something you will easily give kids. But there are some medications in the pipeline that are more gentle. So I think with them, it could.
Sorry. So great talk. Thank you. I have a question regarding the behavioral component that is actually the only behavioral component that is correlated with your brain data, right? It's basically the motor inhibition component that is contributed by a subset of tests, right? And my question is whether the testing itself might contribute to the fact that this is the only component. So what I'm trying to ask, are you asking for response in all those types of, or these are actually not being done during the fMRI?
They're not.
They're not,
outside the
they're outside.
scanners.
Okay.
Yeah, and you're touching a very important point for the reason we're doing resting state and not task-based, because I think in these kids when the performance is affected, so how can you compare? You know, it's not a fair comparison. If I function on one thing in one way and you,
But you do have attention components here that you can actually test, that you can actually test without without any task. We can talk about this.
okay,
Yeah. Right.
yeah, yeah, I like to.
More, perfect.
So I have two quick questions. So one, in the beginning, you mentioned a term called a genetics first approach.
Yeah.
I was wondering if you can comment a little bit about what that is and what's the significance of that. And the second is in the beginning, you talked about the CNVs, the within psychiatric
Yeah.
disorders. And what's the connection between the CNVs and the oncogenic pathways? Is there a connection?
Okay, yeah, maybe I didn't emphasize that enough. So I'll answer the second question first, and then so there might be a connection. I wasn't focusing on that, but what I wanted to show here is that where I think that these mutation on the RAS-MAPK pathway, they actually sit here. They're a single disease mutation, and because it's a single disease, it's a better model in my view, because it's biology. You don't know what's going on, but at least you have one gene to start with, in contrast to a chunk of genes that are being deleted. Now, I'm not, for example, with 22q, I'm not. There's no association that I know with like oncology. The association with oncology in the RAS-MAPK pathway is because it's like oncogenic pathway. I'm kind of like convinced that in some of these deletions, if you delete like a chunk of the chromosome, some of them can be associated with more malignancies, but I have not reviewed that. What was the first question? Yeah. What does it mean? Yeah. So it means an approach in psychiatry, because for many years in psychiatry, we've been struggling because our diseases are very heterogenic. And for example, when you come and you do a earlier work, for example, in brain imaging of kids with ADHD, we'll take 20, 30 kids with ADHD and compare them to controls. Now, probably you're comparing something that is very heterogenic to controls. And then we started to see there are findings, but they're not consistent, and it's really hard. So those studies became like larger and larger and larger, and results come today from really large cohort because of the small effect sizes. So the genetic first approach is saying something else. Instead of increasing, maybe if I'm putting that in contrast, I don't think it's in contrast, but instead of like trying to capture more of the variants of a very heterogenic condition, let's start with a gene that we know what's going on with them. And we can even measure the activation of the pathway. And then, so it's a genetic first in terms of like, let's first think about the genetics and then think about the psychiatric phenotypes, which usually is not available to us, because when a patient presents with first psychotic episode, most of the cases you won't find yes. I have to say, you're small but mighty.
Really interesting talk. I work in a schizophrenia clinic, and I'm interested in, like, a lot of the work in that area. It seems to recently focus on the connection of genetics to sort of immune and inflammatory kind of related. It seems like there's some sort of convergence building there, and complement systems, things like that. do you see any sort of potential connection between this sort of pathway, this oncogenic pathway, and the kind of more immune related theories, or do you think they're really distinct?
So I haven't thought about it yet in the context of the RAS and MAPK pathway, but you gave me a great idea. But I will say that I thought about it in the context of sex chromosome aneuploidies, because we do know that in terms of the immune system, we do have sex differences that are quite significant, right? Women tend to have much more autoimmune diseases, while men are more prone to infections. So I did a review on Turner syndrome and Klinefelter. It's in the Neuropsychopharmacology 2020, I think. And there we did find that the autoimmune conditions that are associated with Turner and Klinefelter are not the same. And that, for example, boys with Klinefelter syndrome that have an excess of the X chromosome are more prone to autoimmune, like females. So I definitely think there's a lot there. Also, kids with 22q have association with, I can't remember now the specific, but they do show a different profile of the immune system. So I definitely think there's a lot to that. And I don't want to even think about cancer and autoimmunity. And yeah, so I'm sure there's a lot.
Thank you so much, Tamar. We really appreciate it. Thank you, everyone who joined us in person and online. We'll log off and say thank you. And everyone here is obviously welcome to continue chatting.